Continuing on where we left off before, so we were talking about Mendel and his experiments on peas and how they were concluding basically different traits of the peas. And then he found out that there's dominant genes or dominant traits that will mask the less dominant ones. They show us an example here of round peas being crossed with wrinkled peas, and all of the offspring become round. So by looking at that, we know that round would be dominant over wrinkled peas. So then we took those round peas and we crossed them with round peas, so we crossed them with each other. We ended up getting a little bit of a different thing showing up. So we got some round peas, the dominant, and then we got a quarter of them being wrinkled. So by doing this experiment, he realized that, okay, there's not just one gene, there's one from each parent, and even though you look like you're dominant on the outside, you might have one of those not dominant genes being masked. So I call that, masked. And there's a word for that, and I don't know if it's going to be on the next slide. Okay. It's 2 slides from now. We'll get there. So our traits, it says they're acquired traits during a person's lifetime because of experiences, education, and upbringing, such as a scar from a cut, of the ability to speak a certain language, or I'm guessing that's supposed to say. So acquired traits are things that you acquire over your lifetime, where inherited traits are traits that are genetically passed on from one generation to the next, such as a certain blood type or eye color. So something that you can't change over your lifetime, it's what you're born with. So on here they've got the word alleles, and I was just looking to see exactly what they're meaning by this, but alleles are basically just different forms of the gene. And when we're talking about peas, for instance, we use letters to represent the different traits. So if we're talking about round versus wrinkled, then we would use different letters basically to identify it, and those letters are considered alleles. So for a round, you can pick any letter really, and the letter I'm going to choose I'll pick R. So a big R signifies that it's dominant. So for round seeds, they could be big R, or they could be big R, little r. And why they could be big R, little r is because this big R right here will mask this little r. So even though on its gene level it'll look like big R, little r, what we'll see with our eyes is round. Now wrinkled, there's only one possibility. If you're wrinkled, you have nothing to mask. So you would have to be little r, little r. So once you don't have that big R in there to mask, that dominant trait isn't there, then you're gonna see wrinkled peas. So these little letters that identify that are known as alleles. Okay, so this is an allele and this is an allele. And the different alleles will signify basically whether you have a dominant or recessive trait. Okay, something that people might sometimes do is they might write, well, this is big R for wrinkled and this is W for round and wrinkled. We need to always keep the same letter. So if we're talking about the same trait, whether you're round or wrinkled, so smooth or wrinkled is really what it is, sort of the texture of it, then you're going to use only one letter, so the big form and the little form. If you start changing letters, then we're going to think you're talking about a totally different trait, like color, for instance, and that's not how we do it. So we need to always keep that same letter. I don't care what letter you use as long as you're being consistent. And normally we just use the letter that represents the trait that's dominant. So round starts with R, we'll use an R. So it gives you an example. Consider the trait like your thumb. One allele creates a straight thumb, while the other creates a hitchhiker thumb. So that's that one that we saw in the last lesson, looking at your thumb or looking at your tongue. Remember that little activity? So if we had a straight thumb as being big S and a hitchhiker thumb as being little s, then we would think that straight thumb is dominant over hitchhiker thumb. And basically you have a trait from your mom and a trait from your dad, and those two together form what you would look like, having a straight thumb or a hitchhiker. Okay, here's some terms. These are pretty important. I'm surprised that they didn't talk about these yet. These are the terms that explain all of the different things about traits. I've already used the word dominant. So something is dominant. There we go. And then the word recessive. So something that's recessive is something that's not dominant. So a dominant allele is something that will show up, the trait will show up. Where a not dominant allele, also known as recessive, you'll show the recessive trait. So I did go over this before, but here if we were going to do this, this would be the recessive traits. This is recessive. And then the other ones are our dominants. And that's what the alleles would look like. So the bigger letters are dominant, the smaller letters are the recessive trait. Homozygous, it says your two alleles are the same, two dominant or two recessive. So what it means to be homozygous is that you have the same alleles. So if we look at these ones above, these are both the same, these are both the same, so those would be considered homozygous, okay? These two, not the same, they have two different letters. They would be known as heterozygous, okay? So homozygous, I always tell people homo, if you know the word homo, it means the same. So homozygous, you could think of homosexual, means you like people of the same sex. If that's what you need to remember that, whatever you need, homozygous, that means the same. Heterozygous means that your two alleles are different, so one dominant and one recessive, and you will show the dominant trait. So even though this one right here is big R, little r, you're going to show whatever the trait is that's big R, because you've got that dominant trait. It's going to mask or overpower that recessive trait, okay? And then the last two here, genotype and phenotype. A genotype is what it looks like at your gene level. So the alleles that you have. So this would be your genotype, and your phenotype would be what it looks like to our eyes. So let's say that's round. If we're talking about peas, the big R signifies round, so big R, little r would mean that it is round. So continuing on, just discuss the different terms that we use for talking about inheritance, and then we're going to move on to how we can actually figure this out. So something that we use to determine the possible genotypes of our offspring from our parents with known alleles is known as a Punnett square. So how do we use a Punnett square? Well, to draw a square, or a Punnett square, we need to draw a square, then label each row and column with the alleles of a gamete. We fill in the square with the offspring's genotype, and then we determine the fraction of offspring with each genotype. This fraction is the same as the probability of an individual offspring possessing a particular trait. So a lot of this is, I have to change over to PDF, so some of it gets lost. So let's try one of these out. So it says our information, one dominant allele is big P, codes for purple flowers, while the other recessive allele, little p, codes for white flowers. It says suppose two pea plants each have the allele big P that codes for purple flowers and the allele little p that codes for white flowers. Let these two plants be the parent generation. Excuse me. Excuse me. Okay, so this is our thing. So they're saying that our parents are big P, little p, and big P, little p. All right. So those are our parents. This is determine the color of the flowers in each of the parent pea plants. So these are our parents. If we go back to the top here, it says our dominant allele big P stands for purple. So all of these parents have big P's, which means this is going to be a purple flower crossed with a purple flower. So it says to set up a chart on Punnett square to show all the possible outcomes of the fertilizing of a gamete from one plant with a gamete from the other. So we're going to set up a Punnett square. I don't know if this is really important, whether we know that these are purple flowers, but what's really important here for our Punnett square is that we have this, okay? So draw our square. Our square is only going to have 4 slots in it. We're going to draw one parent up here, big P, big P, little p. It's hard with P's because it's hard to see whether something's a big P or a little p sometimes. So it's not always the best to choose a letter like this. That's why I like picking R. S is a really terrible one because it's hard to tell. A is a good one. N is a good one. So when we know a difference between our upper and lower case, it's easier to see. So we've drawn this out, so now we need to do the cross. So how we do this cross? Well, we just write, it's sort of like battleship, we write these two in this circle, okay? So it becomes big P, right? Because this is a big P coming from there and a big P coming from there. Then in this square right here, we're going to take the big P from there and the little p from above. Okay. I just want to simplify that a little. So big P from the left and little p from above. And then big P from above, little p from the right, and little p from both the top and the left. Okay, so when we do that cross out, we get 4 possibilities, and this is sort of creating a percent. So we need to analyze this. We need to analyze the genotypes and the phenotypes. We're going to do genotypes first. So genotype is what it looks like at the gene level, and that we can take right from our chart. So I like to group these. We've got this one's big P, These are big P, little p. And this one is little p, little p. Okay? So those are the options that we have, and we just want to write those as saying it's a percentage probability here. So I'll do that. So our big P, what is our percent? Well, it's 1 in 4, which is equal to 25%. Our big P, little p, there's 2 out of 4 of them have become that, which is 50%. And then our little p is 1 in 4, and it's 25, which is 25%. Okay, so that is how we would determine the percentage probability. So if we were going to cross these two parents, the probability that we get a offspring that is big P, little p would be 50%. Okay, the probability of getting that would be 25%, and the same little p would be 25%. Okay, so that's how we figure that out. So letter D here says, suppose the two parent plants produce 12 plants in the first generation of the offspring. How many of these plants would you expect to have white flowers? So we're supposing that the two parents produce 12. I'm just going to do this on the bottom here. So if we're producing 12 plants, they want to know how many are white. Flowers. Okay, so the easiest way to do this is, now that we've got our percentages, we can just convert that to basically how many it is out of 12. So if we have 12 plants, how I would do this, I'm going to possibly, need to write on the next slide, but anyway, so how many are white flowers? Well, if we look at white, if we look at these, big P would make it purple. This is big P, which would make it purple. So in our possibilities, our probabilities, the only ones that are white flowers are these. So 25% chance that we're going to get white flowers. So we can just take our 12 plants and multiply it by 25%. So if you want to do this as 25%, it's best to do it in a fraction, in a decimal. That's what 25% is in a decimal. That's why I wrote it there. And by doing that, we can figure out how many plants are going to be and maybe you can do this in your head, like a quarter of 12. You can probably see it. But if I go 12 plants times 0.25 in my calculator, I get 3. So I was just double checking, mostly because I wanted to make sure that it worked out properly for you. So yeah, definitely a way that you can do that in your calculator is this. So how many of them are going to be white? Well, if we're using the same parents, then we should see roughly 3 of the 12 plants would be white. Okay, so 3 of the 12 would be white flowers. Okay. So now I'm not exactly sure what is going on here. it says homozygous versus heterozygous. So it wants us to do some examples here. So I will try some examples out with you. The first one is a heterozygous parents. So that's the one we just did. But we can do that one again if you'd like. I'll do that one a little bit quicker. So we got big P, little p with big P, little p. So if we do that out as a Punnett square, you get big P, little p, big P, little p. And if we cross them, we will get that. So this would be, what's our possible outcome? Well, our possible outcome is that a quarter of them will be big P, a half of them will be big P, little p, and another quarter will be little p, okay? That's the one we just did, because that's a heterozygous parent. They're heterozygous because these are different. They've got different alleles, sorry. And I used red for that. I like to be consistent with that. So those are our heterozygous parents. If we're gonna do a homozygous dominant parents, homozygous means the same, and dominant means big letters. So that means that our parents are going to be all big letters. So if we do that as a cross, big P with big P, hopefully you can realize that all of our offspring will be big P's. Okay? So the possible outcome, it's going to be 4 out of 4 or 100% big P, which is purple flowers, right? All right. And then the final one here is a homozygous recessive plant. So homo means the same, recessive means the smaller letter, so it would be all little p's. A little bit close. So all of them again would be 4 to 4 of them would be little p, which I did as being green. That is 100%. And that would be white flowers. All right, so that's how you cross these. Just remembering you're using these words, the homozygous, heterozygous. You can always refer back to the definition page. But my greatest way of saying homo, as long as you remember homo means the same, and then dominant and recessive, you'll do pretty well at this. All right. This is my favorite part of genetics, just because it's fun. It's sort of like math-like, and I like math. Okay, so that is that. There's some questions that I'd like you to do that go along with that. All right, so I'm going to continue on. Because, yeah, we'll do as much as we can today. So it says genotype versus phenotype. It says an organism's genotype is a description of the alleles that it possesses. So that's our genotype. So it's all the alleles. It's at the gene level, which we already talked about here. The genotype for tongue rolling tongue rolling couple. Could be, I'm guessing that's what it's trying to say. Could be homozygous dominant, heterozygous, or homozygous recessive. So one of those is going to be the genotype. So genotype really is these letters that we assign to that. And that's only in our head. I mean, that's not really, you don't look under a microscope and see little R's. That's just a way that we represent them. But remember that genotype is that gene level. It's not what we see with our eyes, but what we see microscopically in your DNA. Where phenotype would be either tongue rolling or non-tongue rolling. It's literally what it looks like. So what it looks like to your eyes. Okay, that is that. So multiple alleles and sex-linked traits. It says incomplete dominance is a cross between organisms with two different phenotypes that produce an offspring that has a different phenotype than the parent. It's sort of cut off there, but the point is that incomplete dominance is basically you're creating a third genotype. One gene isn't dominant over other. And in the case of these flowers, red is not dominant over white. They both have incomplete dominance, or they both are at the same level. So then we end up with plants that are pink. So we've got little a, big A, and little a, and then a big A, little a will look pink. We would expect it to look red because we would expect red to be dominant over white. But in these types of cases, they just show up as a mixture of the two, which is really common in life. We often don't see complete dominance. There's a lot of incomplete dominance, especially when it comes to colors of different flowers, different skin tones, different fur colors, depending on what we're looking at. That's definitely incomplete dominance. Oops. Codominance is a cross between organisms with two different phenotypes that produce an offspring with a third phenotype in which both of the parental traits appear together. So an example of this is an incomplete dominance, but they're actually both showing up. So you see this white cow and brown cow, and then you get not a beige cow, but you get a white and brown cow. So that's what codominance is, that both of the parents' traits are showing up. Sort of like a speckle, and you see that in this flower where